And that next speaker is Dr. Kejin Hu. Dr. Kejin Hu is an assistant professor in stem cell biology and cellular reprogramming at the University of Alabama at Birmingham School of Medicine. He completed his PhD program in molecular biology at the University of Hong Kong in 2003. Excuse me. He initiated his career in stem cell biology as a postdoctoral scientist at the University of Wisconsin-Madison in 2007. Since then, he has published more than 10 articles in cellular reprogramming and pluripotency. Dr. Hu is an ad hoc manuscript reviewer for more than 10 scientific journals, grant reviewer for multiple funding agencies, and is currently on the editorial board for the Journal of Stem Cells and Development. Dr. Hu's research is supported by the Alabama Institute of Medicine, the Faculty Development Fund of his current university, the American Heart Association, and the NIH. Dr. Hu?
Thank you, Catherine, for your nice introduction. Hello, everybody. I'm sorry, I have to say, today I have some allergy. If you hear me coughing, just forgive me. My research is cellular reprogramming and pluripotency, especially human iPS reprogramming. Today, I'm going to share with you two stories in my lab. If you have any question at the end, you don't have chance to ask. Here is my email. Just email me any question you may have. So oh, my mouse is not very okay. Now it's working. So today I'm going to share with you two short stories in my lab. But before I talk about iPS reprogramming, so it's a good idea to briefly refresh your memory about the basic of embryonic stem cell. When we talk about embryonic stem cell, we refer to the cell we culture in the lab, isolated from inner cell mass of a pre-implantation embryo. The first embryonic stem cell, embryonic stem cell 9 were established in 1981 by Martin Evans, Gail Martin, and Matthew Kaufman. It took us more than 17 years to make the same achievement in human. In 1998, Dr. James Thompson from the University of Wisconsin, he successfully cultured human embryonic stem cell. Human embryonic stem cells are infallible resources because each cell has two basic features that tissue stem cells do not have. First of all, embryonic stem cells have the potential to become any type of cell in our body. Secondly, we can culture embryonic stem cell in the lab forever. It has unlimited ability to expand. Because of these two features of embryonic stem cell, they have many critical applications. First of all, the greatest promise for embryonic stem cell is cell therapy. Also, embryonic stem cell can be used in drug screening, basic research, and disease modeling. Embryonic stem cells are infallible resource, but embryonic stem cells are not without any limitation. In fact, they have three critical limitations. First of all, not everybody likes the idea to use embryo in the research and clinical setting. Even though everybody is okay with using embryo in the research, we don't have sufficient supply of embryo. To obtain embryo is not like to get a cup of a Starbucks coffee on the street. Thirdly, we have a critical technical problem in using embryonic stem cell in clinical setting. As you can see in this slide, to establish embryonic stem cell line, we have to use ICM, inner cell mass. But it is this ICM that develop to a human being. Using one embryo, we can establish a human embryonic stem cell line or natively develop to a beautiful human being, but we cannot get both. In this case, this embryonic stem cell line is nobody's cell. So if you transplant the cell derived from this ES cell line into a patient, you will have a problem. You have immune rejection. To overcome this issue, scientists have looked for an alternative way to establish embryonic stem cell. In 2006, Dr. Shinya Yamanaka, the Japanese scientist, his lab discovered a new way to establish embryonic stem cell line. His lab successfully converted mouse fibroblasts to embryonic stem cell lines, neck cell. They named this cell induced pluripotent stem cell. Only 1 year later, his lab, using the same reprogramming factor OUT4, SOX2, CMYC, and KL4, successfully converted human fibroblasts to iPS cell. At the same time, Dr. James Thompson lab in Wisconsin also converted human fibroblasts into human iPS cell. They use a different set of reprogramming factor, OUT4, LANOG, and NIN28. Now we can convert a lot of different cells into iPS cell. Here are several examples. We can convert keratinocyte, T cell, B cell, blood progenitor cell, fibroblasts, and neuron stem cell into iPS cell. In fact, iPS cells have the same cell morphology, colony morphology, very similar transcriptome and epigenetic landscape as the ES cell have. Most importantly, iPS cells have the same differentiation potential of ES cells have. Like many other technology, iPS cells, the original technology, also have a lot of room to improve. Even now, in my opinion, iPS cells still have a lot of problem. It is still, this reprogramming is still inefficient. It is a process, slow, incomplete, stochastic. We have aberrant reprogram-associated mutagenesis and transformation. Today, I'm not going to discuss the detail about this improvement. If anybody are interested in the technical detail, I have 2 comprehensive reviews published in 2014. In these 2 reviews, I have discussed many technical details, but today I do want to mention one problem with the original protocol, because iPS reprogramming is a long process, especially for human iPS reprogramming. It takes more than 2 weeks. In this case, we have to integrate the reprogramming factor, these 4 reprogramming factor, into the genome to be reprogrammed. This integrating reprogramming has 3 critical problems. First of all, if you insert any foreign gene into the genome to be reprogrammed, you have a risk of insertion mutagenesis. Secondly, we know these 4 reprogramming factors are silenced upon completion of reprogramming, but much of the time, the silence is not complete. This incomplete silencing is indication of incomplete reprogramming. Also, all these 4 genes, the expression level of these 4 genes have to be in a narrow window. If you have a residual expression, your iPS cell will lose the identity. Finally, even all the transgenes are silenced completely, This gene can be reactivated anytime during the differentiation process or after you transplant this cell into the patient. To overcome this problem associated with the integrating reprogramming approach, James Townsend Lab developed a new reprogramming system. It's called episomal reprogramming. These episomal plasmids do not integrate into the genome to be reprogrammed. This plasmid is very simple. It just needs two components from the virus. One is origin of replication is only 1.7 kb. The other is Epstein-Barr virus nuclear antigen 1. With these two components from the virus, this plasmid can replicate in human cell and also can partition when the cell divides. But this partition is not perfect. However, this is an advantage in terms of reprogramming. We can isolate iPS cell without when in the population lost the plasmid. Indeed, Jun, my friend, she reprogrammed human fibroblasts into iPS cell and could able to isolate iPS cell without any transgene. We use different approach to prove this. We use RT-PCR. We don't see any expression of transgene. And we use genomic PCR. As you can see, there's no integration, no free plasmid. Our southern blot analysis also indicate there's no transgene in this iPS cell. So using this episomal system, we can human fibroblast into iPS cell. However, the expression, the efficiency of reprogramming is very, very low. When I was a postdoc in Dr. Igor Slokvic's lab, we hypothesized progenitor cell might be easy to reprogram. So we use blood progenitor cell because blood progenitor cell are the most accessible progenitor cell in our body. I developed a protocol. I can reprogram blood sample, cord blood, and bone marrow frozen in the liquid nitrogen for more than 10 years. Indeed, as you can see here in this figure, a episomal reprogramming of bone marrow is more than 100 times efficient compared to fibroblast reprogramming. Indeed, I isolated iPS without any transgene in this cell. Now this next slide this slide is a slide I made 7 years ago when I established my own lab. I still believe what this slide states still holds true. I still believe reprogramming is a black box to us because reprogramming even now is still very inefficient. It's a non-process. It is a multi-step process. It is still stochastic. We have reprogramming associated mutagenesis and transformation, and many cells are incomplete reprogramming. About this reprogramming black box, I have two major hypotheses then. First of all, I still believe there are additional reprogramming factors we don't know. And secondly, I believe there are a major reprogramming barrier we still don't know. Now I'm going to share two small stories. First, I tell you how we discover a new reprogramming factor, and then I will show with you how we overcome reprogramming barrier. As I said, I think there are new reprogramming factors we don't know. So to search for the new reprogramming factor, we bought a human Kejin CDA library. Kejin Hu, my postdoc then, he evaluated the reprogramming activity of the human Kejin CDNA. After he evaluated 89 CDNA, he found some candidate, and then he did a secondary screening. He found one CDNA consistently enhanced IPS reprogramming, and then he tested this CDNA in 4-factor condition and 3-factor condition. Indeed, this CDNA enhanced reprogramming activity significantly in both conditions. Based on the library, this is a human BRD3 CDNA. We then purify this CDNA and sequence it. Surprisingly, this is a short isoform of BRD3. Because nobody studied this CDNA before, we named this protein BRD3R, standing for BRD3 with reprogramming activity. So we found a new reprogramming factor. So to understand how this protein enhances reprogramming, we conducted RNA sequencing using RNA from the only reprogramming cell. Interestingly, we found a lot of mitotic genes are up-regulated by BRD3R. You can see in this figure, we found a lot of geoterm contained the keyword mitosis, M phase, and related geoterm. Indeed, after we conducted more RNA sequencing, we found 128 mitotic genes are consistently upregulated by BRD3R. This indicates BRD3R has a mitotic activity. This is confirmed by our additional experiment. Our mitotic shake-off experiment indicated that upon BRD3R expression, we have more mitotic gene. We have a similar result when we use flow cytometry to profile the cell cycle. Now, you know BRD3R has two activity, reprogramming activity and mitotic activity. Does it make sense? Can these two activity explain to each other? My answer is yes. I believe BRD3R mitotic activity is responsible for its reprogramming activity. To explain this to you, I have to introduce another reprogramming approach. This is outside reprogramming. Outside is in fact the most powerful reprogramming feature in the nature. Associated with outside reprogramming, we have another technology, somatic cell nuclear transfer. This cartoon explains to you how this technology works. You remove the nuclear of outside and then take a nuclear from a somatic cell. Most of the time is fibroblast. Then you put the fibroblast nuclear into the enucleated outside and let this reconstructed outside to develop. And then you get a blastocyst. Using this blastocyst, you can do two things. First, you can establish embryonic stem cell as you do using the normal blastocyst. This is called new NT-embryonic stem cell or NT-pluripotent cell, NT standing for nuclear transfer. Using this blastocyst, you can also to clone animal. The most famous cloned sheep, Donny, was cloned using this technology. Here I show to you this mouse is the first mouse cloned using somatic cell nuclear transfer. Her name is Cumulina. Using SCNT cloning technology. Over half a century, we have two very interesting observations. For both the recipient cell and the donor nuclei, we know mitosis is very critical for cloning. In this table, you can see oocyte. Oocyte is a vehicle we use to clone almost all the animal we clone now. But upon fertilization, zygote lost animal cloning capacity. However, Kevin Egan from Harvard University, his lab did an experiment. His lab showed if you arrest zygote in mitosis, this zygote regained animal cloning capacity. Even for plasmid from a two-cell embryo, this plasmid has no capacity to clone animal. But Kevin Egan's lab showed if you arrest plasmid from a two-cell embryo, this plasmid regained reprogramming activity. At the same time, John Gordon's lab, the Nobel laureate, in 2014, his lab published a paper. His lab showed if you arrest donor nuclei at mitosis, these nuclei are 100 times easier to be reprogrammed. Now you may understand why I said BRD3 mitotic activity may be responsible for its reprogramming activity. Now this concludes my first story. My lab screened a human Kylon library, and we discovered that BRD3R is a robust new reprogramming factor, and it might be that its mitotic activity contribute to its reprogramming activity. Now I'm going to shift the gear to another topic, reprogramming barrier. Talking about iPS reprogramming, many people say it is epigenetic reprogramming, it is cellular reprogramming. Today, I would like to use another term, transcriptional reprogramming, because if you want to convert one cell type to another cell type, what you have to do are two things. First of all, you have to establish the transcriptional program specific to the endpoint cell. At the same time, you have to erase the transcriptional program specific for the starting cell. Most of the time, it's fibroblast. Here, I have two heat maps to explain to you these two processes during iPS reprogramming. On the left side, there are a group of genes. These genes are embryonic stem cell specific genes not expressed in fibroblasts, not expressed in bone marrow. So if you want to convert fibroblasts to iPS cell, you have to activate this group of genes. At the same time, on the right side, this is a group of genes, Zolti fibroblast gene. These genes are expressed in fibroblasts, but not in iPS cell and not in ES cell. If you want to convert fibroblasts to iPS cell, you have shut down the expression of this Zolti gene. You may have experience to knock down one specific gene of your interest. You may encounter it's difficult to reach 80% knockdown. So you can imagine how difficult to shut down Zolti gene at the same time. Is this a whole picture? No. How big is the transcriptional barrier to iPS reprogramming? To answer this question, my lab conducted RNA sequencing to compare the expression of human fibroblasts and the human embryonic stem cell, here I show a heat map. As you can see, there are 3,148 genes that are expressed at a much higher level compared to human embryonic stem cell. Among these genes, 1,070 genes are not expressed all in human embryonic stem cell. What does this mean? This means if you want to convert human fibroblasts to human iPS cell, you have to shut down these 1,070 genes and downregulate the remaining more than 2,000 genes. This is a huge task. So how to downregulate these more than 3,000 genes efficiently and specifically? This is difficult to achieve, but today I will offer you a solution. But this is not a complete solution. To explain to you, what is our approach, I have to introduce to you a group of protein, bromine domain extraterminal family protein. Beta protein include 4 members, BRD2, BRD3, BRD4, and BRDT. Beta protein has a unique structural feature. They contain 2 bromine domains at their terminal. Bromine domain has a unique chemical capacity. This bromine domain combines acetylated histone. As you know, acetylated histone is an active transcriptional signal. So because of this feature, we call beta protein epigenetic reader. And also beta protein have another feature. This group of protein can remain associated with mitotic chromatin. I will explain a little bit more later. Because of this feature, we call this protein transcriptional bookmark. Now this slide shows you why we call beta protein epigenetic reader, because they bind to acetylated histone. As you can see, our confocal image shows that beta protein perfectly localized to acetylated histone. Here we use BRD3R and H3K9 acetylation as an example. But beta protein localized to a different loci from the heterochromatin marker HP1 alpha. Our histone pull-down experiment also show beta protein combine to acetylated histone, both for H3 and H4, especially for tetra acetylation. And this next slide show you why beta protein have another name, transcriptional bookmark. I'm not sure if everybody know. In any cell, when the cell are in mitosis, there's no transcription. They don't transcribe. This is called transcriptional shutdown, mitotic transcriptional shutdown. This is very clear if you look at the pore2 localization. Here is our confocal localization of pore2. As you can see, even in anaphase, pore2 dissociated from the chromatin. Because pore2 is not there, there's no transcription. However, beta protein remain associated with mitotic chromatin, as you can see here, from prophase to telophase. This is why we call beta protein transcriptional bookmark. Now you may understand our approach, because beta protein is an epigenetic reader and transcriptional bookmark. And now the literature indicates that bad protein regulate cell type specific transcription and then define the cell identity. Based on this fact, you may have a natural hypothesis. That is, we can block the cell type specific transcription mediated by bad protein. Doing so, we can enhance iPS reprogramming. To do this, the best way may be to use chemical. Fortunately, in 2010, the bad family chemical is infallible. A Harvard lab, they report JK1 is a very good chemical inhibitor of bad bromine domain. As you can see here, JK1 perfectly fits the acetylbinding pocket. On this figure, you can also see JK1 have very high affinity to bromine domain, but bad bromine domain, but has no affinity or very, very weak affinity to other bromine domain. To test our hypothesis, we include bad chemical in our reprogramming cocktail. But this result, you can see, is disappointing. We don't see enhancement of reprogramming. In contrast, the reprogramming activity are impaired. We realize human fibroblasts express BRD2, BRD3, BRD4, and each of these proteins have multiple functions. And BRD4 play a very general role in transcription, that is transcriptional elongation. We just think the concentration we used in the experiment I presented to you in previous slide is a standard concentration in the literature. And we put the inhibitor in the reprogramming cocktail for 3 days. Considering this chemical inhibitor is not member-specific, we think the general function of bad protein may be compromised. And then we treat the reprogramming cell on day 3 and day 5. We remove the chemical on day 4. Interestingly, we found the reprogramming activity are increased even at high concentration, 1,000 nanomolar. Encouraged by this finding, we did a dosage test. We found if we use no concentration of chemical inhibitor, the reprogramming efficiency are significantly increased even for a long treatment, 11 days from day 3 to day 14 of reprogramming. And we found the optimal concentration is 50 nanomolar. This is 10 times lower than the widely used concentration in the literature. We also found at early stage, bad chemical inhibition have the highest reprogramming activity, but later treatment still benefit reprogramming. Also, continuous treatment for 11 days give us the highest reprogramming activity. We also found we use 2 additional bad chemical inhibitor. We have the similar result when we use them at lower concentration. Interestingly, we found during reprogramming, bad chemical also change the cell morphology. As you can see here, fibroblast is have a spindle shape, but in JK1 treatment, the cell become smaller, polygonal. Even some cell become round cell. This is very close to the shape of iPS cell. As I said, originally we hypothesized chemical inhibition can downregulate fibrous transcription and enhance iPS reprogramming. To test this hypothesis, we conducted RNA sequencing. Indeed, when we treat the reprogrammed cell for 48 hours at 50 nanomolar, we found 1,182 genes are downregulated. Of these, 651 genes have much higher expression in human fibrous compared to human embryonic stem cell, and 270 genes are not expressed at all in human embryonic stem cell. So this proved our hypothesis. We also found even in the non-reprogrammed cell, if we treat the human fibroblast for 48 hours, 551 genes are downregulated. Of these, 390 genes have high expression in human fibrous compared to human embryonic stem cell, and 162 genes are not expressed at all in human embryonic stem cell. Now, this concludes our second story. We find a chemical way to erase the fibrous transcription program. Doing so, we can enhance iPS reprogramming. Finally, I would like to thank my lab member, especially Zhichen Shao. He did a lot of work for these two projects. Rather, a good friend of mine, he did a lot of bioinformatics. Also, David contributes for some bioinformatics. Michael Crowney did the RNA sequencing. I also would like to take this opportunity to thank the scientists for offering me this platform to share with you my research. My research is supported by our university and also Alabama Institute of Medicine, American Heart Association, and NIH. Thank you so much for watching my seminar. I'm ready to take any of questions you may have.
Thank you, Dr. Hu. The audience has submitted several questions, so let's get to them. This is a question for both of you, Dr. Stadfield and Dr. Hu. You both mentioned the Yamanaka and Thompson factors used to create iPSCs. Is there much research into using different factors, and are these the ones that are always used, or are there ones that work better?
Do you want to go first? Should I go first? I don't care.
You can go first. You can go first.
I think one of the remarkable aspects of the initial factor combination that Yamanaka described is that it works so well. Okay. Efficiencies are low in some instances, but you will always get your colonies. And for some applications, you don't really care whether it's 1 in 1,000 or higher than that. No. So because it works so well, I think the factors were so successful that really most of the reprogramming that's ongoing is done with the 4 factors. Having said that, there are alternative combinations. I mentioned one of them described by Rudolf Jenish's lab in mouse fibroblast that doesn't use any of the original Yamanaka factors. Efficiencies are lower, but there seem also attractive features of this. Finally, reprogramming has even been reported entirely with chemicals without transcription factors. That could be also an interesting option. But if you look at what is used mostly in also now for disease modeling and so on, it is the classical Yamanaka factors, still simply because they work well.
Yes. I think for both set of reprogramming factors, the efficiency are low, especially for human iPS reprogramming. That's why I look for new reprogramming factor, I believe still we have a very robust reprogramming factor need to be discovered.
Okay, great. Thank you for that answer. This is another question for both of you, I think. Recognizing the complexity of reprogramming, what are the current approved uses of stem cells for clinical therapy?
Do you want to go first?
You can go first.
So the clinical, I will answer this question for pluripotent cells, not for stem cells, because bone marrow transplantation, of course, is a stem cell-like therapy that has been used for decades. Pluripotent stem cells, approved therapies. There are a couple of clinical trials that have been approved in Japan very recently to generate dopaminergic neurons in the brain of a patient. These are pioneering studies. There is no routine clinical intervention that is based on pluripotent stem cell therapy currently. But there has been, for example, another clinical trial that was medium successful to replace retina tissue in 2014, also in Japan. So there are some countries that aggressively pushing these cells into the clinic, but there is no routine process yet to use these cells for cellular regeneration. At least as far as I know.
Sorry, I have energy. Yes, as Dr. Stedfeld said, iPS-based therapy is in the stage of clinical trial already. I have confidence in this kind of therapy, but in my opinion, now reprogramming certainly we need a lot of improvement. We have to develop protocol for a GMP grade IPS for therapy. Also, as I said, there are a lot of a problem associated with current IPS reprogramming, associated mutagenesis, associated reprogramming associated mutagenesis, reprogramming associated transformation, and aberrant reprogramming, incomplete reprogramming. You can imagine for sequencing, in my time, my PhD program, I use manual sequencing, my big deal, but now sequencing, you have a next generation sequencing. For human genome, it took several years to complete, but now 1 week you can sequence the human genome. This is the case for IPS technology. I think for IPS to be used in clinical setting, we need to improve this technology.
I might want to add just one thing here, that there is also, of course, logistic issues with cell therapy using pluripotent cells. And that is simply the fact that you have to, of course, characterize your cells very well. So even if you have a perfect reprogramming protocol, you might have bad luck. You might have started with a bad cell. You might have had a mutation pre-existing. You have intrinsic instability of pluripotent cells even if they have never been reprogrammed. So you have to test these cells. And that actually is interesting because now the clinical trials that are ongoing in Japan, they're actually not using matched iPS cells for this reason. Because it's so difficult to actually, let's say, take a tissue sample from a given patient and then actually make iPS, test the iPS, expand the iPS, differentiate the iPS, and put them back into that same patient. So that is, you can do this as a proof of principle, yes, but to do this as a standard medical procedure is not attractive. So this logistic problem might really might be really the most serious problem for the establishment of cell therapy based on these cells. So I believe that the immediate potential is really in testing drugs, understanding disease. Because here you can discover general principles and find ways of interacting with disease features that are then applicable using drugs for many patients. And of course, that's so much more attractive for companies also than betting on cell therapy. So I am somewhat skeptical, to be honest, about the cell therapy perspectives, at least in the short run.
Okay, thank you. So, you know, you're sort of talking about there's still some logistical issues to overcome. We have another question here. Dr. Stadfeld, I think this will be, we have just time for one more question. Dr. Stadfeld, you mentioned that iPSCs can essentially be cultured forever. Do you know what's the longest time that iPSCs have been grown for, and have there been any issues?
So I mean, every time I make that claim on the slide, I swear to myself I will look into this question and be able to answer it. And actually, I don't know what is the longest. So it's a little bit of a bold statement then, forever. But having said that, even if you can certainly culture these cells for a year or so, and probably somebody has tried this, and I should know this. But there are issues, no? You have one feature of pluripotent cells is that they tolerate chromosomal abnormalities and other abnormalities much better than other cells. Other cells would start apoptosis, would start to die. Pluripotent cells don't. That is convenient, but it's also a big risk, because that means over time you can just accumulate abnormalities in these cells. And indeed, that's what you see. If you grow pluripotent cells for an extended period of time, they will acquire all kinds of abnormalities. Some are neutral, some are actually giving further growth advantage on top of the already impressive growth potential of these cells. I mean, maybe the most famous example is that the second X chromosome in female cells can be lost, in particular in mice. But there are other examples. So long-term culture is feasible, but your cells will drift into undesirable states on different levels, chromosomally, epigenetically.
Can I add something?
Yeah, sure.
Yeah. In terms of culture time, Dr. James Townsend, he established human embryonic stem cell. I have to mention his cell estimated in 1998. His cell line, first cell line, are still used worldwide in almost every lab in this area. Many cells are over 100 passage. Embryonic stem cell in the culture is not stable, but we can isolate a population that is normal. We don't have an experimental result to show how long we can culture this cell, but in theory, we can culture for a long time. Yeah.
Okay, great. So that was a very long time. Thank you. Unfortunately, that's all we have time for today. If you have any further questions, please consider reaching out to the speakers directly. Their emails are shown on the screen. As a reminder, the webinar will be archived on the scientist website, and you will receive an email notifying you when the on-demand webinar is available. I would like to thank everyone who took the time to join us today, and particularly those of you who shared your questions and comments. On behalf of the scientists, I'd also like to thank our speakers, Matthias Stadfeld and Kejin Hu, as well as our webinar sponsors, Biotechne, NanoString, Axol, and Stem Cell Technologies. Thanks everyone, and goodbye.